perhaps go a little bit beyond and give us a vision for the future in their particular field. This year, we are co-sponsoring the event with ACE, the Arizona Cancer Evolution Center, and that's conveniently behind me. That's a major National Cancer Institute sponsored research program aimed at understanding the evolutionary dynamics of cells in tumors and also how cancer has shaped evolution of life on Earth. And you'll find information about both of these organizations in the cards that are being distributed outside. Now, in Beyond Center, our motto is confronting the big questions. For example, a little later this week, we have a workshop on the emergence of meaning. What does that mean? Well, for example, DNA is packed full of coded, meaningful information, the instructions for life. How did such a thing arise in a universe governed by the laws of physics? It's a profound puzzle, but it's a question that nicely brings us to tonight's speaker. If DNA is a type of meaningful text, then that text can be edited, which is the subject of the lecture you're about to hear. Jennifer Doudna is Professor of Chemistry and Professor of Biochemistry and Molecular Biology, and the order of that is apparently very important, at the University of California at Berkeley. Her pioneering work on gene editing technology has opened a vast new world of possibilities and responsibilities, and no person is better qualified to explain the weighty issues involved than Dr. Doudna herself, so I now invite her to come forward and deliver the 2019 Beyond Annual Lecture.
Something called genome editing that came about through a curiosity-driven set of experiments that led in an unexpected direction. Sort of the reason many of us go into science in the first place is that we're fascinated by the world we live in, and we want to understand how things work. And that's always been my reason for doing the kind of science that I do. And I'll tell you a story that began with one set of questions and led in a very different direction. And then I want to turn to the topic of the future. What does the fact that we now have a technology of editing the code of life mean for us as humans and for where we're going in the natural world and what we might do together and to change the world and make it a better place? So let me start with how this technology began. And this is a slide that shows the cell that is being infected by viruses. So this is actually a bacterial cell.
Level is a bit up and down.
Yeah.
It's more reliable.
How's that? I want to start by describing experiments that got started with really just a few scientists around the world who wanted to understand how it is that bacteria fight viral infection, such as this example here, where viruses are landing on the surface of the bacterial cell and beginning the process of infection, and led to led in an unexpected direction to a technology for editing the DNA, the code of life. And this work for me started, if we rewind the clock about 12 years, with a phone call that I got in around 2006 or so from a colleague, Jill Banfield at Berkeley, who works in a very different area of science than me. So I've always studied the molecules of life and investigated what the structures of molecules can tell us about biological function, Jill Banfield has always been interested in the evolution of life. So she studies microbes that populate our environment and sequences their DNA to understand what kinds of organisms are living in our natural world. And her research had uncovered something very unusual, a kind of mystery, and that was that many bacteria, when their DNA was sequenced, revealed a pattern of DNA sequences in the genome. So this is you're seeing a diagram of a circular chromosome of a bacterium here that had an intriguing repetitive pattern. It's shown right here, there we go, where we have repeated elements of about 30 to 40 letters or base pairs in length in the DNA. So these are identical sequences that flanked short sequences of DNA that were unique. And this is an unusual enough pattern that it had been noticed by just a few labs that were starting to sequence bacterial DNA at that time. And they also noticed that these had come to be called Crisprs. Here's it's an acronym for clusters regularly interspaced short palindromic repeats. I won't say that again. and these tended to co-vary with CRISPR-associated or Cas genes. So these were places in the genome that encoded proteins. So this had the look of some kind of a conserved system in bacteria, and the question was why? Why do a lot of bacteria have this pattern? And in around 2005, there were 3 articles published in journals that I certainly wasn't reading, that were all from bioinformatics groups. These were people that do computational analysis of DNA sequences, and they noticed something very interesting. And that was that these sequences of DNA that were unique in these CRISPR elements in bacteria were all derived from viruses. So they came from the viruses that infect these bacteria. So it really had the look of a bacterial vaccination card, a way that bacteria could store pieces of DNA from their invaders in their own genome. And the question was, well, why would they do that? And so the hypothesis that Jill Banfield had at the time was that maybe these were the signatures of an adaptive immune system, a way that bacteria could acquire immunity to the viruses that infect these cells. And she called me because she knew that I'm a biochemist. I've always worked on molecules, and in particular, molecules that control the way that genetic information is used in cells. And so she wondered if we might like to work with her lab to investigate whether, in fact, this immune system was operating, and if so, how it worked. And so what happened over the next few years in research that was done in a handful of labs, and largely initially in work done at a yogurt company by scientists who wanted to have a very practical goal, which was to protect their cultures, yogurt, cheese, things like that, from being infected by viruses and being destroyed, is that it was shown that bacteria that have these CRISPR-associated genes can acquire pieces of DNA from viruses and then generate a copy of these sequences in the form of RNA. So RNA is a chemical cousin of DNA that provides a kind of a little transient copy of that piece of genetic information in the cell. And these molecules of RNA were then processed. They were chopped into shorter bits that each included a sequence in the RNA that comes from the virus. And then importantly, they combine with proteins encoded by the CRISPR-associated, these Cas genes, to form RNA Guided Complexes, where the RNA is effectively the zip code for the protein. It provides the address label that tells this protein where to go. And the way that works is that these little molecules with their RNA guides will search the cell looking for matching sequences of DNA, sequences that match this copy in the RNA element. And when that match occurs, the protein is recruited to that piece of nucleic acid and it gets cut up and destroyed. So you can see how this is a great pathway in bacteria. It allows cells to acquire immunity and then use that information. It programs the cell to recognize those invaders if they should show up again and destroy their nucleic acid. And so
Jill Banfield, my colleague, so we began meeting regularly and talking about this, and my lab started to investigate how these molecules actually function in the system. And meanwhile, Banfield's lab was continuing to do what they do well, which is to research the microbes that populate the environment. And this is a slide that was taken pretty recently where they were, so these are graduate students in her lab. Looks like they're having a pretty good time. And they should go out and get a little dirty, and they, if they collect samples, this is collecting samples of groundwater and soil up in Northern California. And here you can see a trench here with some water in it, and little test tubes on either side. So these are samples that are being collected from different spots along this rope that's laying alongside this little water inlet. And then what's in the samples? Well, this is a diagram of one of these test tubes, and what is done is to filter the water through a filter that separates the organisms living in those environmental samples from everything else. And then they can be easily sequenced. The DNA can be, the code of life in these cells can be read, and all the contents of those genomes can be analyzed. And in the course of doing this type of research, Banfield's lab and others doing similar types of experiments have uncovered many different varieties of this Crispr adaptive immune system, such that we now have a large collection of diverse examples of these pathways. Now, for somebody like me that's always worked on molecules and we study kind of the how questions in biology, how do things work? This is being like a kid in a candy store, right? There's so many fascinating variations on this theme of adaptive immunity. So we continue to investigate some of the molecules that are part of this pathway. And this is actually a cartoon that illustrates some of the diversity that's observed. So we can separate these CRISPR systems into two different classes, and beyond that we have different types. These on the top here in class 1 require multiple genes. That's what each of these boxes represents that are required for the immune system to function, versus the types shown down here called class 2 that each have a large gene with a single protein that's encoded that is essential for the function of the immune system, which is kind of interesting diversity. And back in 2011, we were initially investigating these class 1 systems with their multiple proteins and trying to figure out how they worked. And then I went to a conference and I met a scientist named Emmanuel Charpentier, a microbiologist who was studying a different kind of system belonging to the class 2 type down here. When we met at that conference and we started talking about our lab's respective research projects, we realized that we could team up to figure out the answer to what seemed like just a very natural question at the time in the field of CRISPR biology, which was, what is the function of a gene known as Cas9, one of these large encoded proteins that is essential for function of a CRISPR immune system in certain types of bacteria. We were drawn to this because it seemed amazing that you could have systems that in some cases required many proteins to do what in this system it seemed like only one protein was needed to do. I thought, well, that's got to be a pretty interesting protein, kind of like a Swiss Army knife that might have lots of different functionalities to it. And so I wanted to understand how it might work. Now that embarked our labs on a wonderful collaboration that allowed some young scientists who were working with us to do some really fun experiments. And in particular, we started investigating the way that this Cas9 protein is able to cut DNA at places in the DNA sequence that match this sequence in the guide RNA molecule. This is showing a diagram of this. So this protein turns out to be a chemical cleaver that can literally make a double-stranded break in DNA at a site that is marked by the sequence in the DNA matching this sequence of letters in the RNA guide. So when that double-stranded break occurs in bacteria, this leads to destruction of the DNA. And so, of course, if this RNA guide is recognizing a piece of viral DNA, then the cell can cut it up and destroy it. But when Martin Ginnick, the postdoc in my lab that was doing this work initially started investigating how this works, he realized that, first of all, this is a protein that's guided by two separate types of RNA that are shown here in the cartoon, CRISPR RNA and a type of RNA called tracer. He figured out that these could be linked together into a single RNA molecule that we call the single guide RNA that would have the structural handle needed for interacting with Cas9, so that would be the same on every RNA molecule in the system. But it was then very easy to change the sequence here so that this address label could direct Cas9 to any desired segment of DNA. And for us, this was kind of the moment when this project went from being a curiosity-driven question, how does this immune system work, to realizing that this could be a powerful tool for manipulating all types. The cells respond to double-stranded DNA breaks in their genomes differently than in bacteria, where bacteria will quickly degrade the DNA. In our cells, those double-stranded breaks are recognized, and the cell can either, or DNA, if it has some sequence in the DNA that match the sequence elements flanking the double-stranded break site. So if you think about this, it's a way to trigger alterations to the genome of a cell by introducing a double-stranded break at a position where you might like to make a subtle change in the code or make a much larger insertion of new genetic information. So that wasn't our idea, but once we had discovered a molecule that had this very simple way of introducing double-stranded breaks at any desired position, we realized that that molecule could plug right into these pathways very easily and create a powerful tool for altering genomes. I'm going to show you a short video that just illustrates how we imagine this system working in eukaryotic cells. So this is, here we are zooming into a cell, and in our cells, animal and plant cells, of course, the DNA is packaged inside the nucleus, and it's wrapped up around proteins in green here that are form chromatin, so they really compact the DNA into a small volume. And amazingly, this bacterial protein Cas9 with its guide RNA is able to search through all of the DNA in the genome of a cell to find a position that matches the RNA guide. When that match occurs, the DNA is cut, and then this protein hands off the broken ends of the DNA to repair enzymes in the cell, come in, and this is the little bit of the hand-wavy part of the video, right? You sort of imagine how this might work. But in that example, there's an actual insertion of new DNA sequence in the process of repairing the break. And so remarkably, we published this work in the summer of 2012. And we proposed Cas9 and its guide RNA as a technology for genome editing. What happened next was truly remarkable, because laboratories around the world quickly began adopting this strategy for genome editing in all kinds of biological systems, everything from human cells and stem cells to zebrafish, to plants, rice, wheat, to mice, etc. Basically, every kind of organism that scientists were studying in laboratories became a subject for this technology. And this is, in fact, this is sort of over the last 7 years. When I think back on, what's happened over the last 7 years, it's been truly exponential growth of the adoption of this technology. This is actually a slide I got from a publisher, the Elsevier scientific publisher site website. They have this slide that just shows how earlier technologies for engineered proteins that could generate double-stranded DNA breaks and trigger genome editing. These were technologies known as zinc finger nucleases and meganucleases and talon proteins. These all had started to be adopted by the scientific community, but they hadn't really taken off. They were, difficult enough to use that most scientists didn't have either the ability or the funding to, start using these widely. And the difference with CRISPR is that this is a technology that is so easily adaptable, and it's easy to use, it's not expensive, you can train a starting student in the lab to come in and start using it to edit cells within a matter of a few weeks. And so it took off like a rocket, right? And so this is really showing exponential growth. Now, like I recently did a search in PubMed, which is a web server that allows you to search all of the biological and biomedical scientific literature, and there are literally almost 13,000 publications now, just since 2012, that are using this technology across sort of all different kinds of biology. So it's been a remarkable, remarkable few years to watch this taking off and think about all the things that are now possible using a tool that allows precise alteration of genomes. In my own lab, we've continued to investigate how this works. We'd love to understand this. So I showed you the video, and that's kind of an artist's imagination of how this works, but we'd love to actually understand this better so that we can figure out, first of all, how an amazing little machine is able to find and cut DNA so precisely, but also how we can better understand both where it came from, how did it evolve, and also how we can employ it for things that we might like to do, given a tool that provides this kind of power. So I was going to mention just a couple of things about understanding this molecular mechanism. So we've been fascinated by this question of DNA recognition. How does this enzyme Cas9 gain access to the DNA in a cell? And how does it find the right place to cut? And what happens when it finds the wrong place? How often does it do that? And how does it tell the cell, or how does the cell figure out how to repair the DNA? A lot of these questions really haven't been fully addressed yet, but I wanted to give you a little sense of how we study these sorts of things. This is actually a 3D model of the Cas9 protein with its guide RNA. Here's the orange RNA molecule. And this is a model that was printed on a 3D printer using coordinates that are based on an actual molecular structure of Cas9. So we can purify these molecules in the laboratory. This was actually work done by my former, now postdoc, Martin Ginnick, who did those original work on experiments on Cas9 in our lab, and was able to crystallize Cas9 bound to its RNA substrate. And so what you can see here is that when the protein holds on to DNA, so here's the beautiful DNA double helix passing through the protein, at the position that matches the 20 letters in the guide RNA, this protein pries apart those two strands of the DNA and forms a helix of RNA and DNA inside the protein. And that's the actual recognition mechanism. That's how it finds the right place to make a cut. And then once that interaction happens, there are two chemical cleavers in the protein that can swing into position and actually cut these two strands of the DNA. And I wanted to show you, here's a little animation showing an enzyme that is also, an RNA-guided protein, part of the Crispr pathway. It's holding on to double-stranded DNA, and here you can actually see the different parts of this protein. It almost looks like a clamshell or a baseball glove or something. It's holding on to the RNA guide here inside the enzyme and prying apart the two strands of the DNA so that the enzyme can get at them and actually make a cut. So one of the questions that we'd like to understand better is, what are the energetics of that process? And if you think about it, if we have DNA that's base paired, so we've got two strands of the DNA double helix, and the chemical letters in the code are, of course, paired, A's with T's and G's with C's. And then, as I showed you in that video, DNA in eukaryotic cells is packaged as chromatin, it's highly compacted. How does this enzyme actually get at the DNA and pry apart the strands? And we don't fully know the answer to that question yet. And one of the curiosities is that, like in this cartoon, the enzyme it sort of is able to somehow massage apart those strands. It doesn't have an external energy source. It doesn't, for those of you that are students of biology, you know that a lot of times energetic reactions that require energy in biology use ATP or GTP hydrolysis to drive those reactions, but not Cas9. So where does the energy come from to melt apart two strands of the DNA? Now we suspect that one of the drivers of this is actually the conformational shape of the enzyme and the way that it changes shape when it interacts with RNA and DNA. And I'll show you a little animation that illustrates this. This was an animation made by another student in the lab, Ben LaFrance, based on some work by student Sam Sternberg, cited here, who was able to track the enzyme in different states of its structure as it assembles with RNA and DNA. So this is an animation that starts off with the structure of Cas9 by itself, rotating into position into the shape that it forms when it binds to this guiding RNA. So you saw a big structural change in the gray part of the enzyme that creates a channel down the center of the protein. And when DNA binds, there's an additional change in the structure to accommodate that RNA DNA is the actual mechanism of recognition, and we think that's part of the action of the enzyme that pries apart the DNA strands. And then finally, this part of the enzyme here, the yellow part of the protein, swings into position as a cleaver and is able to cut the targeted DNA. So it's literally a little machine that's designed to grab onto DNA, pull apart the strands, and cut it at a place that matches the letters in the guide RNA. And it's amazingly accurate. It's not perfect, but it's very good at cutting DNA just at the desired place. Because over evolutionary time, bacteria have selected for a protein that would be able to do this quite accurately to protect from viral infection without somehow damaging the genome of the bacterium itself. So I want to now turn to what's happening with CRISPR as a technology. And I want to tell you a little bit about 3 different areas where there's exciting advances happening. And I'm going to focus on some things that you might not have heard about with CRISPR biology, but where I think there's going to be exciting impacts in the not too distant future in public health, in agriculture, and in biomedicine. Then is that it's been possible to use the CRISPR-Cas9 system for gene editing in farm animals and animals that are important biomedically, such as pigs, for the purpose of altering their genetics in ways that could be beneficial. And this to alter the genomes of piglets to make them potentially better organ donors for humans. And this has been done in 2 ways. One is using gene editing to remove integrated viruses that are part of these pig genomes that could if the organs were donated to a person, they could infect people, so we'd like to get rid of those first. And the other thing is to make the organs of these animals more human-like, so that they are potentially going to be received better by a patient that might be receiving one of these organs from a donor pig. And so this is work that's been going on in both companies and academic labs. And this has led to thinking about how you could use gene editing more broadly to impact public health. And this is an area that has taken hold among people that think about the spread of genetic disease, especially by vectors like mosquitoes. This is a slide that I wanted to show. It's actually taken from Science News because it's illustrating something called a gene drive, which is a way to introduce a genetic change into an entire population of organisms using a tool for genome editing. And this had been theorized about for a long time, probably ever since, certainly maybe before I was a grad student, but I remember in graduate school people talking about this. But it never became really feasible to do it until there was an efficient way to alter genomes. And once CRISPR came along, it really became possible then to do this. And the way these work, this is showing on the left-hand side the normal pattern of inheritance. So we have a breeding pair of mosquitoes, and they've got 2 different genotypes. And so you can see that this trait is passed along in a kind of a traditional sort of Mendelian way. But over here, if we have a gene drive in the genome of this organism, then when this trait is passed on to progeny, the trait is passed along together with the molecule, in this case Cas9, that's able to insert the trait into other animals. And so you start to see that over time, the trait can spread and eventually become a trait that's shared by virtually all of the animals in the population. So why do we care? Well, this is something that people are very excited about because it could provide a way to control mosquito populations and in particular control their ability to spread parasites and disease. And so there's been lots of interest in this, but of course it also raises some very important questions about the safety and frankly the ethics of using a technology like this in this fashion in animals or organisms that might be released environmentally and could have unintended consequences. And that's a theme I'm going to keep coming back to, by the way, is thinking about the awesomeness of what we can do with gene editing, but also the responsibility that comes with it, because it is, in fact, a very a powerful tool. I want to turn to agriculture. So this is an example of some work that has been done by a remarkable scientist at Cold Spring Harbor Labs, Zach Lippman, who's been working on tomatoes and other things. But this is a really interesting work that he published about 2 years ago now, where he showed that you could use the CRISPR-Cas9 gene editor to alter tomatoes so that they would bear much heavier crops than from the starting variety. How did he do this? Well, he was able to figure out how to dial up the expression of a gene that controls the number of flowers that are made by these plants. And so without altering anything else about these tomatoes, he just made plants that could bear many more of them. And a little admission here, don't tell anyone, but I'm actually growing these right now at my house. And I'm really excited because I'm going to see how many tomatoes I get this summer. And I'm going to really do the taste test and see, I've got tomatoes growing that are pre-CRISPR and post, and I want to see do they really taste the same. So we'll see. I'll come back and tell you about that. But this is raising something else that I want to point out, and that is that it turns out that this gene that's affected here in tomatoes is found in many plants. So it raises the possibility that we could dial up the yields of other types of plants as well, and do it without altering their other traits, and also do it quite quickly compared to traditional plant breeding. Now this was work that was also published now maybe 3 years ago. A group at Penn State, so another academic lab, was able to show that they could basically knock out a single gene in mushrooms that prevents mushrooms from turning brown when they get cut open. Who knew? So it's kind of an interesting thing. And this attracted some attention because it raised the question of how do we think about a gene editing tool like this with respect to regulation? Are these plant and agricultural products going to be considered genetically modified organisms, GMOs, or not? Well, right now, the U.S. Department of Agriculture has weighed in on the mushrooms. This is an article about these mushrooms. And they decided that in cases like this, where there's no foreign DNA introduced into the mushroom, it's simply a genetic knockout, that these would not be regulated here in the United States. So these are not going to be regulated, versus what happens in other countries. So in Europe, for example, any agricultural product that is the subject of manipulation by a technology such as CRISPR-Cas9 right now is proposed to be considered a genetically modified organism. So we have this funny situation where, depending on the country you live in, something could be GMO or not, and it's raising a lot of questions, and people are trying to grapple with, how do we handle a new technology like this? Is this something we want to encourage scientists to be using or not? And how are companies that might want to develop products with this technology going to be marketing their products in different parts of the world? So those questions are very much ongoing. And then finally, I want to turn to biomedical applications, and I wanted to share one experiment with you. This is actually from our own research. So one thing that the gene editing technology of CRISPR does is it takes something that used to be really hard to do, and it makes it easy enough that even labs like mine that never in the past worked on, certainly barely worked on human cells that you could grow in a lab dish, much less whole organisms, we started thinking about opportunities to investigate how you could use a gene editing technology in the brain. And this is research done by a postdoc in my lab, Brett Stahl, who has been investigating how you might deliver the gene editor, CRISPR-Cas9, into the brain and get meaningful levels of genome editing that might one day allow us to do things like cure neurodegenerative diseases that result from genetic mutation. And this is an experiment where he chemically modified the Cas9 protein to give it cell-penetrating properties. And then this can be injected together with its guide RNA into the brain. This is doing an experiment in a mouse brain where we inject this editor into the brain, and then we can actually follow cells that get edited because this mouse has a label in the genome that gets turned on. It makes a red protein only when gene editing occurs at a precise position. And so what you can see here is that when we inject on two sides of the brain, we actually get a decent volume of tissue that's edited on both sides. And when we look in detail at what cells are being altered, we find that these are neurons, and we're getting editing at exactly the position that this Cas9 protein is programmed to recognize. And right now, we're actually using this in a mouse model of Huntington's disease, which is a neurodegenerative disease caused by a single gene that's been mutated. And we're trying to figure out if we can actually use this strategy one day to create a treatment or maybe even a cure for diseases like that. That's a long time down the road. That's, in my opinion, the kind of thing that academic labs should be investigating. This is not going to be prime time for a company soon, but it's a great thing to be studying in the research lab and trying to figure out how we might do this kind of thing safely and effectively one day. And then I also wanted to point out that in addition to using gene editing molecules to do things like this, it turns out that in research that was done by a couple of graduate students in my lab, Alex East-Seletsky cited here, and Janice Chen and their colleagues cited here, these students figured out that there are some additional biochemical functions of CRISPR-Cas9 proteins, some of them, that actually allow use of these proteins to detect nucleic acids, to detect DNA and RNA molecules precisely. And they can detect these molecules by turning on an activity in the protein that releases a fluorescent signal that we can read out very easily in the lab. And it doesn't require any amplification using the polymerase chain reaction. We don't need fancy equipment. And we can get very sensitive detection of particular sequences. So, why would you want to do that? Well, it turns out that this can be very effective at detecting the presence of infectious bacteria, viruses. We can also detect the presence of sequences that are antibiotic resistant in these organisms. And in the future, we're hoping we can actually use this to detect things called single nucleotide polymorphisms, or SNPs, that might be associated with either genetic disease or even cancer. And this has led to a whole sort of growing field of use of the CRISPR-Cas enzymes for diagnostics, and the idea that one day we may have simple tools like this that we can use in a point-of-care setting, in a clinic, maybe a very low-tech kind of clinic out in the field, where we can easily tell people that they have an infection that needs to be treated, or that they have some genetic predisposition that they need to pay attention to. And finally, I wanted to turn to the question of how genome editing, so turning back to the use of CRISPR-Cas tools for genome editing, how this editing is actually performed, because it turns out that this leads to another profound question about how gene editing will be applied in humans in the future. And to explain this, I need to point out that fundamentally there are two different kinds of cell types that we can edit. There's somatic cell editing, which is what most of the biomedical applications happening right now are trying to do, which means making genetic changes in cells that are not heritable, so they're not passed on to future generations, and they affect just one individual. But we can also imagine, and we do, germline editing that leads to heritable changes in DNA. And these changes affect not only an individual, but also all of their offspring. Now, this kind of genome editing started to be done very, very early in the in sort of the applications of CRISPR-Cas9 technology, with the use of germline editing in mice. This is actually this is showing a mouse, fertilized mouse egg. It's being held by a pipette tip over here on the left, and you can see a needle coming in on the right and injecting the CRISPR-Cas9 molecules into the cell, where genetic changes that are made then become part of this entire organism. And when this mouse is born, those changes are part of all the cells and can be passed on to future generations. So, of course, you know, when this was done easily in mice and then in rats and then in zebrafish and other kinds of organisms, people started thinking, gee, I wonder if that could be done in mammals and could it be done in humans? And so back in 2015, you know, I'd been sort of thinking about this, and there was work that was published earlier in 2014 by a lab that showed you could do this kind of germline editing in monkeys, and it worked very, very efficiently. And so the question was, you know, how long will it be until someone does this in human embryos? And that question really bothered me a lot. And so I actually organized a meeting in early 2015 with about 20 scientists who met out in California to talk about this question. And what do we do as scientists? How do we grapple with this technology that puts an incredible capability really within pretty easy access of people that know what they're doing and know how to work with embryos? And so that led to the publication in the spring, I think it was early 2017, of a report by the National Academies of Science that really looked deeply at this question of human genome editing, and in particular human germline editing, and made a set of recommendations that included the recommendation that this kind of editing not be used clinically, meaning to create a genetically edited person, until and unless the technology could be thoroughly vetted, as well as to provide time for societal discussions about the ethics of using this technology in that fashion. Now, if you were reading the news last November, you might be aware that there was an announcement in November about someone in China who actually decided to go forward with clinical use of human germline editing. This is a picture that I took of this scientist. His name is He Jiankui, and he came to a meeting that I was part of. I was on the co-organizing committee of in Hong Kong, and announced that in fact he and his team had made genetic edits to 2 baby girls that were born with changes to their genome that were designed or intended to alter a single gene that is involved in susceptibility to HIV infection. Now, this was a really kind of a watershed moment. It was for me, really kind of a crazy experience of seeing this thing that had been imagined for a while, and we sort of hoped that these guidelines that were adopted and put in place by an international committee would be respected. But it was remarkable to see this announcement, because as the data were unveiled for this set of experiments, it was clear that, in fact, what this scientist had done was to conduct experiments on people in a way that I found really quite shocking. And I'm going to show you, I just want to show you one slide. This is maybe my wonkiest slide. I'll try not to get too technical here, but I just really want you to notice, all you have to notice here is that the lines don't look the same. So the top is a cartoon of this gene, which is known as CCR5, and this indicates the position adjacent to the site that was edited. He wanted to make a change, wanted to introduce a change into these baby girls that would replicate a natural occurrence that's very rare in human populations, but creates a deletion of 32 base pairs in the DNA of this gene that protects people from HIV infection. It's one of the ways that this gene, which encodes what's known as the HIV co-receptor protein, the way it was actually identified. But when he used the gene editing technology in embryos, this was done in the vitro fertilization clinic, the changes that were actually made, which are shown down here, don't look like this, right? So these changes that were actually introduced and then identified by DNA sequencing are changes that have, to our knowledge, never been observed in human populations, and furthermore, they've never been tested in animals. So this is truly experimentation on people. And so I think this really, for many of us, certainly for me, it really highlighted the need to accelerate the discussions around this and to really think hard about how we put in place regulations, requirements, whatever you want to call it, that will, we hope, avoid future use of technology like this, that, where the outcomes here are unknown. We don't know what will happen. We hope these baby girls will have healthy lives, and but it really is unclear right now what the consequences will be of the genetic changes that were introduced into their DNA. Now, there's also a lot of hype around Crispr babies, and this was actually a picture. This was on the cover of The Economist magazine at least 3 years ago now, under the banner "Editing Humanity." And there's been a lot of speculation about, could we have perfect vision? Could we have high IQs? Could we cure baldness? Something that some of my friends think would be pretty cool. The answer is that in most cases, the answer is no, because we don't know enough about the human genome to do these things, even if we wanted to. And in many cases, these are traits that require probably tens or maybe hundreds or even thousands of genes that interact subtly with each other. And so they're not going to be easily altered by a gene editing tool that is maybe at most able to alter a handful of genes in a cell at a time right now. Nonetheless, I think we really have to be thinking hard about what's going to happen down the road. And for the students here, in the audience, this is something that will undoubtedly affect you in your lifetime, is thinking about how people will use gene editing in the future. And in particular, will there come a time when people are using germline editing in embryos to make alterations to the genome that they think are going to be beneficial in some way to their kids? It's a question, but I think it's a question that's not that far beyond where we are today with this technology. And so I think one very positive outcome of the announcement in Hong Kong in November is that the World Health Organization has recently announced the convening of an international forum to look into the editing of human genes. And in particular, they are starting to make very vocal sort of recommendations that would establish, I don't know if you saw this announcement recently, but they want to actually have a registry that are trying to use germline editing clinically, so that it would be public knowledge who was doing this. I think that's one thing that we came away from the Hong Kong meeting understanding, is that we don't want to see this kind of thing going on in secret. It should be public knowledge. And then furthermore, I think there's a push, and the National Academies of Science, who I've been working with, will be announcing soon sort of a similar kind of parallel international forum that will be working with the WHO group. And the idea there is to establish some very clear requirements for any future use of clinical germline editing in humans. And that could establish a platform for regulation if countries would like to do that. But at the very least, it would be, again, a very public set of criteria and requirements that would need to be met for any future groups or teams that want to do this. So I want to just wrap up here by pointing out that this RNA-guided ability to recognize a position in a genome, if you think about it, is a really remarkable tool, because I didn't go into all the details tonight, but because we can now direct these proteins to places in the genome, we can actually not only induce gene editing, but we can also change the levels of proteins that are made in cells, like that example with the tomatoes. And we can also do things like image the genome. We can light up particular genes and look at where they are in the cell and how they're interacting. So it's a very powerful platform that's driving a lot of science right now. A lot of the publications that are coming out are just making new versions of this and using it in all sorts of creative ways. We know that Figuring out how to deliver gene editing molecules into organisms and tissues is going to be critical for all kinds of applications, not least of which are biomedical. And also figuring out how to control these gene edits, not only in a chemical sense, so how we induce the cell to make the kind of edit that's desired, but also how we figure out societal controls and regulations that will ensure responsible use of the technology. And then, of course, ongoing fundamental research continues, and that will, I think, continue to drive not only this technology, but what comes next, whatever's after CRISPR, right? And I personally think that a good bet of where that'll come from is from the kind of work that Jill Banfield and her colleagues are doing, where they're out mining these microbes in our environment to find out all of the interesting biological pathways that they have that haven't even been investigated yet by scientists. I'll conclude there. I want to acknowledge a great team. So one of the things that I love about doing science is that I have the privilege of working with incredibly smart people who come to the lab. These are graduate students, postdocs, undergraduates who come to work on projects and work together with each other, but also with various collaborators. So we've worked with a number of colleagues at Berkeley who are shown here. Of course, Emmanuelle, I'm always very grateful to her for the work we did originally with her lab on CRISPR-Cas9. And then increasingly, we're working with colleagues like Joel Pilecki and his lab members at UC San Francisco to apply CRISPR tools in the clinic. So with his team, we're actually developing these CRISPR-Cas enzymes for diagnostics to detect cancer-causing viruses in tissue samples that come from people that are living in various parts of Africa where they don't have access to really high-tech tools, and so having a technology like this could be very beneficial. And then finally, when you're an academic scientist and you're doing research, we're all dependent on people and organizations that provide the funding for this type of work. And we're grateful to all of these organizations, but I particularly want to call out the Howard Hughes Medical Institute and the National Science Foundation. So these two organizations gave my lab critical funding in the very early days when we were curious about this possible bacterial immune system called CRISPR and had no idea where it would go. So I'm just really indebted forever to those organizations for trusting us, letting us do the curiosity-driven science that led to the work that I showed you today. And I'll stop there, and I'd be happy to address questions. Thank you.